That's great. Thank you, and good morning. I'm Pete Vallone. I'm the leader of the Applied Genetics Group at NIST, and we're in the Biomolecular Measurement Division up in Gaithersburg. So today I'm going to talk to you about human identity testing using DNA. So some of the topics, the things I'd like to talk about today are just what is human identity testing? It's also known as forensic DNA testing. So that's kind of the context of what we'll be talking about. And I'll kind of go through the process of how it works, how DNA typing works, a little bit of genetics, and hopefully that'll give you an idea of how human identity testing works and answer some common questions about it. From there, we'll move to sort of NIST role, what we're doing at NIST in terms of supporting the forensic community in human identity testing. And then we'll kind of finish up with some of the newer things that are going on in terms of the future of human identity testing. So if you have any questions at any point, just feel free to stop me. So this is my team in the Applied Genetics Group. We're working in forensic and clinical genetic areas, and this is the team here. The folks on the left are most affiliated with some of the forensic projects that we'll be talking about, but they're a great team, and most of this work is generated through their hard efforts. So starting off, I wanted to just talk about some general characteristics of genomic DNA in relation to human identity testing. Some of the things here, I would say that each individual has a unique DNA profile, and the one exception to this might be identical twins. They'll have a very identical profile. I have here a little up at the bottom, a kind of a little reference down here, and there is some work that's going on with sort of higher level sequencing, full genome sequencing, that's showing that you can actually differentiate between identical twins, but that's not, it's sort of at a finer level than we're doing for human identity testing. Another thing here, each person's DNA is the same in every cell. So the idea, if I have your blood or your saliva or whatever your cells are, you should have the same profile or same barcode in that DNA. So if I get DNA from your hair, it's going to be the same as DNA from your blood. There's a consistency there. And another thing is that your profile remains the same throughout your life. So if you're typed when you're a newborn or 10 years old or 50, you're going to have the same DNA profile. And maybe you could think about this, in terms of maybe fingerprints might change over time or things like that. And then in terms of inheritance or genetics, half your DNA comes from your mother and half from your father. And this is pretty straightforward, but we'll show some interesting examples of this or how that bears out in testing. So then in this case, it has implications for determining kinship. So if you think of other methods of identification like fingerprints or iris scan or facial, those don't really have that type of ability to determine kinship. That's sort of unique to DNA. So here I have just a map of the human genome. You have 23 pairs of chromosomes and then something called mtDNA, which stands for mitochondrial DNA. You have 2 copies per cell. So here I have chromosomes 1 through 22. So you have 2 copies of each of those. So you have 2 chromosome 1s, 2 chromosomes 2. And the one I have here on the bottom right are your sex determining chromosomes. So if you're a male, you have an XY. So the Y is the male specific. And then if you're female, you have an XX. So these are typed, or we'll show how these are typed in human identity testing so you can tell the sex of an individual. I'm not going to say a lot about mitochondrial DNA. It's also in the cell. It codes for different functions. It's much smaller. So your full genome is about 3 billion bases. Mitochondrial DNA is a circular genome. It's about just under 17,000 bases. But there's hundreds of copies per cell versus 2 copies per cell. So sometimes this can be used for a highly degraded sample where there might not be much of the autosomal DNA left, but there's some of the mito left. And I'll talk a little bit later about sort of the inheritance patterns specific to mitochondrial DNA. So what we'll be talking about are what we're calling these autosomes, or these sort of main batch of chromosomes here. So kind of a couple high-level statements about human identity testing. One thing we're not doing to say is we're not sequencing the whole genome. So you have these 3 billion base pairs, and when you hear about a DNA test for, like, say, forensic purposes, we're not going through and sequencing all that DNA. What we're really doing is probing subsets of genetic variation in order to differentiate between individuals. So we look at about 13 to 20 markers or so throughout the human genome, and I'll show you where those are located. So you're looking at this really small subset that allows you to have a high power of discrimination. So this is kind of sometimes a misconception. People think that you're sequencing all their genes and have a lot of sort of disease-related information in a forensic test. That's not true. DNA typing must be done efficiently and reproducibly. So this information must hold up in court. There's protocols and there's standards and guidelines for doing DNA testing. Right now, there's over 13 million profiles that are in the national FBI database. These are all done with similar techniques, similar standards and guidelines to do that. So you have really this harmonization throughout the US of how the typing is performed. You don't sort of have individual or rogue laboratories kind of doing their own method of testing or looking at their own markers. It's all very systematic. Also, too, this is changing, but really we're not looking at genes. So the regions that we're targeting lie outside of genes, so you don't have any, let's say, ethical issues with knowing something about a disease state from an identity test. So there's really not much information about ancestry, things like eye color, hair color, facial features in the standard test that we'll be talking about. But this is evolving over time. So right now, I'll show at the end of the talk, but we're starting to look at different areas, looking at prediction of eye color or hair color, which you could think could be useful sort of in a cold case where you have a DNA sample but you don't have a suspect. But right now, that's not commonly done. And at the bottom here, I have the FBI website. You can kind of, if you go to this website, you'll find up-to-date statistics on how many profiles are in our national database. Right. So this is really simplistic, but just one way to think about it, that there's you, and then you're made up of cells which contain your chromosomes and your DNA. And within that DNA, there's a unique identifier or barcode inherent in your DNA. So if you shed cells or you leave blood behind, that barcode is being left behind, and it could be potentially or traced back to you if there's a reference sample. So if you just have a DNA sample and you generate a profile from it, let's say it's off a coffee cup or a cigarette, you can't say who that person is unless you have that person's a reference in a database or you have a suspect that you're checking. So the information itself is limited unless you have a reference or unless you have something to compare it to. So that's just kind of just a simple concept, but just if you're leaving materials behind, that can be traced back to you if you're typed again or if you're in a database for a reference. So some of the common sources of biological evidence, sort of the ones here on the right, sort of like blood, saliva, and semen are really common in forensic cases, that blood is left behind. It's easy to get DNA profiles off saliva, so if you took a cotton swab and swabbed your cheek, that would be enough DNA for a test. And then oftentimes in sexual assault cases, there'll be semen left behind mixed with female blood or fluids. On the left here, these might be more common sort of in a limited case where you have a tooth or a bone. Think of something like a mass disaster, like a plane crash, or something like the World Trade Center. These are a little bit harder to get DNA off of, but there is DNA in here. And here on the bottom left, I have hair follicle. If a hair is ripped out, there's usually some cells on the end of it, and that's useful for typing. Often, if it's just a shed hair without the root on it, it can be difficult. There's not enough DNA in the hair itself. So the idea here is just really only a very small amount of blood is needed for a DNA profile. So if you had, this is just a piece of paper and there's a dime for reference, but even this really tiny spot here, you could punch that out, put that into a tube, and develop a DNA profile off of that. So in essence, you really need a couple hundred copies of the human genome to perform one of these tests. They're quite sensitive. Some of the applications, and we've talked about a couple of these, but I'll sort of just go through them. Obviously, I think the main one that people know about is forensic cases, so the idea that you're matching a suspect with evidence. So you have some evidence, you've typed it, you have a suspect that you've typed, you see, is there a match there? Or you take that evidence and you search it versus something like the national database, and you see if there's a match there. Paternity testing, so if you have an alleged father in a case, you would type the child and the mother and then type the alleged father, and I'll show an example of this later, but see, does everything match up there in terms of genetic identity? Missing persons investigation, so if you have remains or people that are missing, if you have their relatives or you have a reference sample, you can identify them. In terms of a military dog tag, when someone goes into the military, what they do is take some of their blood, and it's usually on a piece of paper. They kind of prick their finger and bleed out onto this card, and that's stored. It's not typed, but it's stored, so if anything happens to that person, that card can be pulled and they can match tissue or that person's remains with the DNA that's on that card. So I just wanted to mention that because I think a lot of times it's thought that the military has all these samples typed. That would cost a lot of money, and there might be some also privacy issues. They're not, that sample's not typed unless something happens to that individual. And then we talked about the National DNA Database. So convicted felons are swabbed, and those are sent to the FBI, and those are put into the database. We said right now there's about 13 million samples in that database. So this is also moving towards arrestees in certain cases where you're arrested. You can also be enrolled into the database as well. DNA typing is used for mass disasters, so like we said earlier, something like a plane crash or something like the World Trade Center, where you're counting for remains. So the idea, if you have bone fragments or tissue and you're typing that, you might have reference samples like a toothbrush or a hairbrush from someone that you could match to a reference, or a reference could also be a family member, and that's where the kinship kind of falls in. So if someone's in a mass disaster, if they have a brother or sister or parent, that could be useful to sort of re-identify or reassociate those remains. And then also things like historical investigations. You might be aware of things like sort of the ancestry of Thomas Jefferson and or the Russian Tsar. These types of DNA tests can also be used to sort of put back together a picture of a historical question or nature. And then also what's common too is genetic genealogy. So people wanting to trace their ancestry through something called lineage markers. And we'll talk a little bit about those to sort of say what lineage did they come from throughout time. So I'm going to start to get into the actual DNA typing. And one thing here is, what type of genetic variation are we looking at? I said that we're not sequencing everyone's genome, and that's what I have here on the top is just sequence variation. So the idea here, these are called single nucleotide polymorphisms. And what I have here is that in black and underlined, that would be a conserved sequence. So we would all have the same sequence there, but this would be I have here a G and an A. So one individual might be a G, one might be an A. That's a way to differentiate between individuals. And then you would go back to the sort of the common sequence. I think there's about 3 million of these SNPs, or single nucleotide polymorphisms, in the human genome out of 3 billion base pairs. What we're going to talk about more in focus today is called length variation, or short tandem repeats, or STRs. And this is really the workhorse of identification. So similarly in black and underlined here, you can see you've got a common sequence, and then what you have is this repeated motif of GATA, GATA, GATA. So that repeats 3 times, and then you go back to your common sequence. So we'll get into this in a little bit more detail, but really what you're looking at in human identity is how many times does that motif repeat at a certain part of the genome for an individual. So one way to think about it is a kind of an accordion-like sequence that occurs between genes. And again, I have it here where the sequence in black would be conserved between 2 individuals. And then this repeat motif, it's what's going to be variable. The number of repeats there is going to be variable. So in this case, you can count these off, and then there's 12 GATA repeats. So in a test, the number 12 is what's reported, and it's called an allele. That's just the state of that marker. So you can think about it in a population of individuals, the number of consecutive repeat units can vary between people. Or the reason that these markers were selected in the first place is because they are shown to be variable in a population. So one person at a specific area of the genome might have 7, someone else might have 10, someone else might have 13, and so on. So the idea is that the frequency or the occurrence of those repeats or those alleles observed in the general population, they've been sampled, the science has been done here, and then they're used in the statistical representation of a DNA profile. Okay? So taking this sort of just a little bit further, the idea here, let's say I was going to test one of these markers to see how useful it was, and I test 100 individuals, and I would write down how many times I observed someone having 7 repeats, 8 repeats, 9 repeats, and so on. So I kind of have just a mock example here of really the most common would be the 10 repeat allele, and then the least common would be 13. And you could look at that, you can convert to the frequency by dividing that out by 100. So you could see 5% of this population would have 7 repeats, 2% would have 13 repeats. And then another way you could look at that information is taking 1 over that number and saying 1 in. So for the 7 allele, it's a frequency of 5%. So that means that the occurrence of that is 1 in 20 in the population. And sort of the extrapolation or the science that's been done here is that this level of sampling has been performed with hundreds of samples to get these different frequencies for these markers. So the idea here, if someone had a 13 repeat, what's the chance of randomly picking someone out and saying that they would have 13? It'd be 1 in 50. So I'm kind of going through a little bit of detail here because this is sort of where you get your really large numbers that you might hear about on TV or court, 1 in a billion, 1 in 5 trillion. This is where this comes from. So I mocked up another example, and I just want to take you through it. So the idea of the marker here is just that I'm looking at 13 different markers, and they're all independent from one another, so they might be on different chromosomes. I've left out what the allele number is right now, but I just put down a frequency. So the idea here, this is 20%, so it's 1 in 5, this might be 4%. So once you type someone, you get these different frequencies for these alleles, you assign those, and since these are independent events, you can multiply them, something called the product rule. You're just multiplying these through. And so on the end, for the full profile, you can get an idea of what's the frequency or what's the commonality of that specific profile in a population. So it's 1 times 10 to the negative 10. It's very rare. And if you flip that over, that's where you get a number like 1 in 6.7 billion. So this is an example, and a statement that would be used in court is the chance of randomly selecting an unrelated individual having this exact same profile is 1 in 6.7 billion. So that's where these numbers come from, these really huge numbers that you hear about. It's really from taking these frequencies, multiplying them together, and then getting a frequency of that profile and taking 1 over it. So, and you know, the research and science has been done behind these really in the mid 90s, late 90s to kind of confirm that the number of sampling and what's been done has been sufficient. So this is where these large numbers come from, and you could see if you go to court, you would say what's the chance of someone else having this profile, or what's the chance of you pulling someone randomly out of the population having that same profile. That's the number they would give in court for that information. All right. So I put up a picture here of the human genome again, and what I'm going to point out first are the core markers that are used in the United States right now. So these were set in about 1997, 1998. There's 13 regions that are typed in every forensic DNA test, and these are all listed up here. Usually the name has to do with what chromosome they're on. So D3 is on chromosome 3, D5 is on chromosome 5. Some of them have more historical names like TPOX, FGA, things like that. So when you're doing a human identity test, there's commercial companies that make the test for this. All these markers are in that test. And when you upload something to the FBI database or you're doing a court case, you have to have these 13 markers attempted in your typing. So right now, we're in the process, the US is in the process of increasing the number of markers that they're using. So the ones I've highlighted here in green are now going to be added to the 13 to equal 20 total core markers for the United States. So in the next year or two, labs doing DNA typing will have to submit all 20 of these markers to the database. So the reason for increasing them, I'll take your question in a second. The reason for increasing them is giving higher levels of discrimination. You can see here also the sex typing marker this is called amelgenin, but you can know if someone's a male or female. And then also in the past, we didn't have a lot of harmonization or overlap with Europe. So by adding some of these markers, they're also common in the European database as well. So it gives us sort of a more international harmonization of the information. And just overall, having a core is useful. So every lab in the US that's doing testing is testing the same information, and you can share that in a database. So there was a question?
You said the markers are independent.
Yes.
All 13 of the markers are independent events.
Yes.
How do you know that? I mean,
well, there's genetic tests that you could do with family samples and see that the independence means is a state at one marker associated to another marker. And in genetics, typically, if something's on a different chromosome, there's not an association. So something on chromosome doesn't have sort of a linkage to something on chromosome 2. But that's done with genetic studies using family samples and statistics to look and type multiple samples and see is there any kind of correlation between a 7 allele here and a 10 allele here. So there's metrics for that, and those types of studies that would have been done. But sort of in terms of physical distance, if something's on a different chromosome, it's considered to be independent. But you do studies with samples to show that there is no linkage or correlation between the states. And that allows you to do this product rule, this multiplication of frequencies. Does that answer your question?
Yeah. Is the error rate in determining the number of repeats, is that essentially 0?
I would say yes. So the idea of something 7 repeats, you don't really still see it in error with that. It's reviewed by an analyst. We'll show it a little bit later, but there's a ladder that helps size that up for you. There might be errors in interpretation, potentially, but usually there's not an error associated with that sort of identity of 7. You don't say that's plus or minus. There's like a 5% chance it could be an 8. There's a 4% chance it could be a 6. It's usually set as a ordinate or identity information.
Hey, Pete, what happens if all the markers are not available in a given sample?
So if you get a partial profile, so let's say you're attempting 13 and you only got 6 to work, you would have a lower match probability, so it might be 1 in 1,000 or 1 in 10,000. And what you would, in that sense, you would sort of be limited in that sample. So if you weren't able to type it, you would get limited information. So you could think of a forensic case where maybe someone touches something, you try and type it, you only get 5, let's say 5 markers to work. It only gives you a probability of 1 in 10,000. That's just the limited information that you have out of that. So, yes.
And a sort of related question. In the example where you showed the uniqueness looking at the 13 markers, does everyone look at 13 markers? And if so, why are 13 markers selected?
Well, everyone is looking at 13 markers right now. They're going to be going to 20, like I said. The 13 were chosen really sort of, if you think about the mid-90s, you didn't really have the Human Genome Project yet, so there was really limited information that was out there on these markers. So these were the ones that were sort of being discovered at that time, and then it sort of the idea of at some point you have to set a core to kind of get a database going, so that was just a selection in time.
And they said human genome has been entire. When they say the human genome has been entirely sequenced, does that mean that you know exactly where the variations are and where they're the same between all people?
Trying to think. So you're asking if you've done the sequencing of, like, if they've sequenced the human genome. And if I want to make sure. Right. I mean, it's limited in the sense you have a reference genome. So the one that was sequenced it is specific or limited in that it's one individual or a pool of a couple individuals. So it doesn't account for all of it. Sequencing a genome, what's in, let's say, NCBI, which is the data bank for that, it doesn't mean that it's accounting for all human variation. But now that some of the sequencing technologies are getting cheaper, more and more individuals are being typed, but it's really a starting point to have 1 or 2 composite individuals. So it doesn't necessarily mean that from that information you know all the variation that exists.
Do you have a sense of, between any 2 individuals, what percentage of the genome remains the same?
I think it's right. It's highly conserved. It's highly conserved, I would say.
Would you mind just repeating?
The question was how much is conserved, let's say out of the 3 billion base pairs, how much is conserved between individuals? I believe that number, like a simple number would be like the 3 million SNPs that I talked about where there's variation, so it's really a small percentage of the 3 billion. There's some other types of higher level variation or structural rearrangements in the genome, but a simple answer would be about 3 million out of the 3 billion vary within the human population. So I think we're done with this one. I just wanted to get the point across that we're moving towards more markers in the United States for higher probability of matches and harmonization with Europe. What I wanted to get into now was just the actual lab work or the steps in forensic DNA analysis, so you can get an idea of what goes on in a test. And I've broken this down in these different steps of collection, extraction, quantitation, PCR, and then separation and detection. And I'll talk a little bit more detail about PCR and separation and detection. So the first thing you do would sample the evidence. So it might be a swab, a cheek swab if it's a reference sample. It might be getting blood off genes, something like that. So you would sample that and put that into a tube for extraction. So the idea, you're putting cells into a tube, but the cells are what contain the DNA. So what you need to do are break open the cells with chemicals and liberate the DNA. And usually you degrade the proteins that are there, so you end up after extraction with a nicely purified sample just containing DNA. Post that, you don't know how much DNA is there. Is there too much? Is there too little? So you do something called quantitation, and that's where you get an estimate of how much DNA was recovered. And that's important because in the next step, the PCR step, and we'll talk a little bit about PCR in a minute, you need to have about a nanogram or about 300 or 400 copies of DNA to put into this PCR test. And that's what that does is that you have genomic DNA, but now what you're doing is amplifying just the regions that you're interested in. And I'll show how that works in a minute. So you have your full genome, but now what you're doing is just putting in targeted sequences to help amplify the STR regions that you're interested in. You also label those with fluorescent dyes for detection. And then what you do is put this on an instrument called a capillary electrophoresis instrument, and you separate these fragments. And since they're all different sizes, they're going to have different mobilities. And that's what these, we'll show a better version of this, but these peaks here on the bottom are what an STR profile looks like. So in the end, you have these different peaks, and then you assign back the repeat number to those, and you review that profile. So this is kind of an overview. This pretty much can be done in a day with current technologies. So briefly, just if you don't know what PCR is, it stands for polymerase chain reaction. And what it is, it's discovered in the early to mid 80s. It means to create billions of exact copies of a region of the genome. So the idea is like if you only have a genomic sample, you really can't see that under a microscope. You can't tell what those repeats are. What you need to do are sort of amplify those specific repeat regions. So the way this works here is that this little instrument here is called a thermocycler. So all it does is it heats and cools. It's like a kind of an oven. This would be your tube that has your DNA in it and your chemicals or reagents for PCR. And then this would represent, this would represent a helix right here, your region of DNA. And DNA is double-stranded, and it's not, if you heat it up, it breaks apart. So it's held together by weak forces. So if you heat it to almost boiling, at 98 degrees C, you have what's called denaturation. The two strands break apart. And then what you have is you have a cooling, and you have these things called primers. And what those are regions that are outside or just, they sort of bound the STR repeat region. Those stick to the DNA that you just denatured. And then you have what's called a polymerase and sort of the A, G, C, and T, the different chemical moieties that are in there. And what happens is the polymerase, using the template that you denatured, makes a new strand. So if that makes sense, you're basically breaking these apart, binding primers, and then each of those strands that have been broken apart make a new double helix. So you repeat this over and over. So you have exponential amplification. So the idea of if you start with 2 copies, after the first cycle you go to 4, then you go to 8, 16, 32, and by your 30th cycle you have over about 2 billion copies or so. So this has kind of been, really in the 80s, this has really revolutionized the area of molecular biology and biochemistry. So this is what the profile for heating and cooling would look like. You have this heating for 1 minute. This lets the primers bind for a minute, and then the polymerase makes the new chain or makes the new piece of DNA for a minute or so. And these other incubation steps are for enzyme activation and incubation here. So, and then it's kind of restating, as you do more and more cycles, this is how many copies of that region that you end up with. So if that makes sense, that's what you're doing when you're doing this PCR reaction. You're basically amplifying what you want to look at in this reaction. And at the same time, you're also labeling these fragments with a fluorescent dye for detection. And that's what I'm moving to here, just talking about separation and detection of PCR products. And this is done with capillary electrophoresis. So you could think of this right here, this tray, as your samples. You have these very thin tubes that have a polymer inside of them, and you apply a high voltage. So what happens is these fragments migrate through the capillary, and then at some point, after about 36 centimeters or so, you have a fluorescence, a laser that hits them and detects the dyes that are attached to the fragments, and then that signal is what's collected as your peaks. So the simple way to think about it is something that's, let's say, 20 repeats is going to come through slower than something that's 10 repeats. And you have sort of internal standards or markers that allow you to do the sizing and assign the values. So this is just another way of looking at this would be a kind of a cross view of 4 capillaries, and these different fragments with the different alleles or different repeat lengths go through the tubes and are detected by the laser. This takes about an hour or so. So in the end, you'd end up with your profile. And really, these peaks that are now labeled with the alleles on the bottom here, they represent your DNA profile that would be unique for an individual. This is the capillary electrophoresis instrumentation. It's about as wide as this podium or so, maybe a couple feet tall. And then this is what we're talking about here. This is where the sort of the buffer solutions, right here is where the laser will strike the capillary. And then this is a 24 capillary instrument. So your sample tray would be down here around A. You have these 24 capillaries, and that's where the DNA migrates through to be separated and detected. So here's an idea of what these look like just a little bit closer. So this kind of instrumentation was commonly used in the 90s. It's still used today for sequencing of the genome, but also this is what we're calling this is fragment separation, looking at these PCR fragments. So what I have here, this is one of the newer tests I talked about that we're going to 20 markers. The name of this kit is made by the Promega Corporation. It's called PowerPlex Fusion, and this is what a profile looks like. So in the end, after your extraction, your quantitation, your PCR, your separation and detection, this is what you end up with. So there's 4 different colors here. Those are 4 different dyes that the fragments were labeled with. That allows you to sort of multiplex, looking at these multiple markers, and then there's different things you can pull out of the profile itself. So this would be a nice profile. This is a male, so you have an X and Y peak here. And then what I'll do, since this is kind of hard to see, I'll just blow this up a little bit further. So let's just see, at this locus here called D2, this individual on one chromosome has 17 repeats, and on the other chromosome has 23 repeats. So this is deemed heterozygous. There's a different state on each of their chromosomes. Whereas on D21 here, it's a 28-28. So each of their chromosomes has the exact same repeat allele, if you will. And then here D7 is an 8-11, D19 is a 13-13, and then FGA is a 22.2/25. So this is what an analyst would do. They would look at this profile, look at these peaks. There should be really no more than 2 peaks at a specific marker because you only have 2 chromosomes. If you had 3 or 4 peaks at a marker, you would think you have a mixture. And that would be common in a forensic case because you may have a mixture of 2 people's blood or fluids, things like that. So this is essentially what a profile looks like. Now, when we talk about putting these into a database, we talk about matching these, we're not talking about taking these peaks and sort of trying to overlay them with another profile. Really, all the information is distilled down to the allele calls. So for each of the markers that's on the left here, or loci, you have the state. So it's a 15-15, 11-13. This is what gets put into a database, and this is what's searched. So if that makes sense. You're not actually searching these peaks. You're just kind of, it's really a flat file, a text file, that's being uploaded to the database and being searched. But in that sense, the profile's gone through double review, usually in a forensic laboratory, and these are confirmed here. So, yes.
Fractional counts? How do you get a fractional repeat?
So it's fractional in the sense that, okay, so there's 4, most of these are called tetranucleotides, so it's a GATA, or it's some 4-base repeat. Well, in some cases, you might have a motif that one of them, due to mutation, might go to 3 or it might go to 2. So the 22.2 means there's 22 4-base repeats, and then there's 2 more bases. So one of them got truncated by 2. So if it was 22.3, then it'd be 3 bases. So it's called a microvariant, but it's just it's a sort of a diversion from the normal tetranucleotide repeat. So going through with actual data here, we can multiply the frequency of each of these genotypes that are on the left here, and that gives us a random match probability of on the order of 10 to the minus 39 for unrelated individuals. So the idea of randomly selecting an unrelated person out of the population, having that exact same profile is, I don't know what the words would be for something 1 followed by 38 zeros, but it's an extremely, extremely high number. So this is an idea with these new tests, you're kind of going from the idea of 10 maybe to the 13th or 18th to the 10 to the 40th or so. So it's a high probability of a low probability of someone having that exact same profile. So are there any questions on this? This is really kind of this, I wanted to take you through quickly how this is developed, and you could get a feel for what kind of information is in a human identity test. So briefly, in terms of CSI, you can see that you can't perform these tests as quick as a commercial break. But the instruments you see on TV are real. They don't work as quickly as on TV. But this kind of just segues a little bit into some of the things we're doing at NIST in my group in terms of rapid PCR. So that PCR step where you're doing that amplification, that takes about 3 hours or so. Now it's about 2 hours, but it was about 3 hours about 2008 or before there. So some of the work that I did in my laboratory was working with faster polymerases. That's the enzyme that makes the complementary strand, and looking at faster heating and cooling methods. We reduced this from 3 hours down to about 36 minutes. So we kind of published this to the outside, and some of the commercial entities like Life Technologies, Thermo Fisher, and Promega have taken that on. And now the commercial kits that are out there now, there's one that works in about 40 minutes, and some of the other ones have been brought down to about 90 minutes or so. And this is also supporting something we'll talk about in future technologies, is really there's a development of fully integrated rapid DNA instruments. And we'll talk about those in a few minutes, but that's really putting a swab in a box or an instrument, hitting the go button, and then having to develop a profile hands off in about 2 hours. So this is the idea of some of the technology that was worked up at NIST that's had an effect in the DNA typing community. And here's an example of a profile that we generated in our lab. So instead of this taking 5 hours, this is an example of taking a cotton swab of some cheek cells from an individual, and the idea of taking it through all the steps and using a 14-minute PCR that we developed, we were able to get a profile in less than an hour or so. So this is a nice, this is something we published and put out there for the community to help them sort of optimize their protocols and methods for rapid DNA typing. I wanted to show an example of kinship, because I think it's interesting, or it kind of shows the relationship there between mother, father, and child. So you can use DNA profiles to estimate a probability of kinship. And one thing to remember, using the types of markers that we're talking about, as a relationship becomes more distant, sort of the confidence in confirming that relationship becomes it decreases over time, over relationship distance. So the idea of doing a parent offspring, you could do that with a high certainty. So when you do a normal paternity trio where you have a child, a mother, and a father, DNA works quite well. If you don't have the parents but you have two siblings, you can still do a DNA test. Usually that's can be a bit weaker evidence. And as things go out to half siblings, and a half sibling is also defined as uncle, nephew, relationship, or grandchild, that's half sibling. Those become more difficult to have a good certainty on in terms of a relationship. And then cousins is even another degree out. So the further you get away from the parent-offspring relationship, the less information is shared between those individuals. So it makes testing a little bit weaker. So the idea here would be that if you had two parents and a child, that would be an easy test to do. But if you're comparing a brother and sister, in this case they're not identical twins, obviously, but that relationship's a little bit weaker. So the more individuals you have in a pedigree, the more you can do in terms of associating them together with a high confidence. So here's a nice example, a paternity example there's the father and the mother, and you could see up here from this example, I'm just showing a few of their markers, not all 13 or 20. It gets kind of complicated to see, but these two people luckily are not related. You can see that they have different markers. There's an 18 here, there's a 14, 17 here. So at most of these, there's a different allelic state. So if you have the child, and what you can look at here is what we were saying is kind of illustrates you get one from your mother, one from your father. So the idea is you can match up here which allele or which allelic state you got from your mother, which one you got from your father. So in the first case here, it's easy to see that the child's a 14, 18, so that means he had to get the 18 from his father and the 14 from his mother. In this example here, the father was a 6'6", so both of his chromosomes had a 6. The child is a 6'7", and then the mother is a 6'7". But since the father doesn't have a 7, he had to get the 7 from his mother, the 6 from, 7 from the mother, 6 from the father. So you can kind of go through there and see that the child is always a composite of the two parents. And you can put statistics on this, and I'm not going to do that right now, but you can imagine if at one of these markers it was completely different. If the child had, or if the father had, basically if the child had a genotype here, like 20-25, but let's say the father was a 28-28, then chances are that would be an exclusion. So the simplest way to look at paternity is the child share an allele at every marker with the parent. Yes?
So that's a graph of the mom, child, and dad. If the dad was a twin, an identical twin, how could you tell which one is the father?
You wouldn't be, I would say you wouldn't be able to unless you have a lot of money to kind of do more genome sequencing. It would be it wouldn't be done sort of under the normal guise of human identity testing. It would have to go through high throughput sequencing. You'd have to look at more of the genome to try and find differences between the twins and then try to associate those with the child. So it wouldn't work well.
It wouldn't work well?
No.
So if two twins died, say in a car crash, would you be able to tell which twin, one's name is Antoinette, one name is Annie. Can you tell which one is Annie and which one is Antoinette?
I would say, well, is that male female names, or those like if they're both the same sex, if they're identical, I would say with this type of testing, no, it would not work.
So I wouldn't be able to know which one of my daughters it is.
No, you'd have to do I would say you have to kind of go to more of like a research genetic type testing way to do that. Yeah, it wouldn't work with sort of these commercial kits.
Okay.
Yeah.
Oh, thank you.
There's another question up front.
How come on that chart, the mom, dad, and child all have a 6, and the first 6 you show comes from the dad, not from the mom. But then over to the right, you have a 12, all 3 of them have 12, and it shows it coming from the mom and the dad. Why is that inconsistent?
You mean this one here?
Yeah, that's one from everybody.
Right.
And then go to the left, and there's a 7. Go to the left more, right there, a 7. And the 6. The 6 comes from the dad.
Right.
But it doesn't come from the mom.
Right.
How come?
Because the child has a 7, and he had to get the 7 from somewhere, and the 7 came from the mother.
The 7 comes from the mother, but the 6 comes from the dad. How come the 6 didn't come from the mom?
Because she gave him the 7. There probably is maybe a 1 in 1,000 chance that her 6 could have mutated to a 7, and that could potentially be the source of that. But
I see. Dad has a 6 and she has a 6, and the child ends up with a 6.
Right, but where does
Each parent can only get one.
so the child has to get a 7 from somewhere. And I guess I may even flipped it. Yeah, the dad would have to have a mutation for one of his 6s to turn into a 7 for that to be the case. Yeah.
Answer my question. Obviously, these things have to mutate, or they would never have evolved this diversity. So,
Exactly.
what is the chance that there would be a mutation in one generation?
It's usually 1 in 1,000, approximately. So if you do enough studies, if you look at enough family samples, you will see a mutation. Yeah, and it doesn't mean from a 1-to-1 matching standpoint, you'd be fine because you're going to be what you are. But it would maybe affect a paternity test where maybe they figure at 20 loci of those 20, maybe at 19 it's a perfect match, and maybe at one of them it's off by 1 unit. So there's statistics to account for that. If it's off at more than that, then the probabilities just drop from there.
Is that the same for all of the markers?
Yeah, for STRs, it's common to say that it's about 10 to the minus third, so 1 in 1,000. But if you do more and more family generations, you'll see where these mutations happen. But you're right. The reason that these are polymorphic from an evolutionary standpoint is that they have been expanding over time.
Can you comment on the signal-to-noise ratio? Because I almost look at the 11-12 in the child, I almost want to say they're 11-12, but I see it's low enough that you probably just say that's noise in
These
the
two
child.
peaks here?
Yeah, exactly. Looking at, that you have the dad as 11 and 12 and the mom has 12 and 13, when you make a call that that would have been 11, 12 in a child, where the 11 came from the dad and the 12 came from the mom, as an example.
Right. Yeah, I can see what you're saying. There's also there's an artifact called stutter. So when you have a 12 allele, or any allele size, usually you have a very small peak that might be like 5%, because when that polymerase chain reaction happens, that has a little bit of error too. So if you could see all these sort of have that, where the 18, this is at 17 right here. You have a little bit of that. But there are I'm not going to get into it, but there are interpretation thresholds. So things below a certain level, you do a validation, and so you know what your signal to noise ratio should be and whether to count something or not count something. But yeah.
Why the 17, which you see way on our left, and the 17 up on our right, was not transferred? What does that mean?
It's just Mendelian genetics. It's the probability. You have the father has 2 chromosomes, the mother has two chromosomes. So you could have 4 possibilities of what could be transferred. So sometimes it is, sometimes it isn't. And that's just Mendelian genetics in terms of the probability there. One thing, we'll keep taking questions, but one thing I was going to throw up, this is the brother and sister. So that's the brother again, and then there's the sister. So I wanted to show here is that, and this might kind of lead into your question a little bit, is there's just these different probabilities of donating these different alleles. So it's interesting, you could see for brother and sister, at this locus, they're both 14 18s. At this locus, they're both 6 7s. If you look at this next one, the brother's a 9-10 and the sister's an 11-14. So they share one allele in common, but the brother got the 9 from the dad, but then the daughter got the, I'm going to confuse myself, but the daughter got the 14 from the dad in this case. And then it's kind of interesting, there's also a probability of having the brother and sister not sharing anything, where the brother here is a 20-25, but then the sister is a 17-21. So they'd be like, I think it's a 25% chance of that. So if you think just a little bit about what I talked about, as a degree of relationship gets out further, the probabilities go down a little bit. So because you have cases where you do have a perfect match for sharing, and there's cases where you don't have any match. So if you just looked at one locus, let's say for paternity test, which you wouldn't do, and you looked at this one here, the 20-25 and 17-21, you'd say they're unrelated. That's why you have to look at more markers.
Thank you. Is there any research for triplets or any greater number of siblings? Or, I mean, you said identical twins. You said share, but what about triplets? Or
if they're identical triplets, they would share it as well. I don't know if there's any specific research. And that one paper I mentioned early on, because the twins and the identical triplet, those questions come up a lot. And I would say 10 years ago, I would say, well, you can't differentiate between them, and especially using this type of marker system, these STRs. But we'll talk a little bit about next generation sequencing, which is high throughput sequencing. And now that you have the ability to do a full genome, you can maybe find, out of those 3 million SNPs, maybe you'll find 5 or 6 that are different between the twins or triplets. It's due to mutation. So the mutation rate for a SNP might be 10 to the -8th. So it's maybe 2 to 3 times less likely to mutate than an STR marker. I believe so. I have to think about that more to answer that properly.
I don't know if you've mentioned it. You probably did. I was wondering about dominant and recessive genes and how this factor into it.
Yeah, I don't. So I'm not a geneticist by training, but I don't believe these fall into that category. They're pretty much, you have an equal chance of donating any of those, the 2 from each parent. So one does not dominate or one's not recessive, and these aren't phenotypic traits, so these aren't predicting eye color or hair color or anything like that. So, but I don't want to kind of go off there too much. I'm not a geneticist.
You mentioned about mutation. When does this happen in development, and are there people doing experimenting with affecting that mutation?
I would assume that it happens at the time of, like you said, the egg splitting or conception. I don't know as much about it. It's not happening, later on in life. It's happening at when the sperm and egg meet at that meiosis point. But again, I'm probably not the expert in that area, so I don't want to expand on there. But it's
experimentation and impacting that mutation selectively.
I mean, for STRs, it's probably not really studied because they don't code, so they're sort of neutral. They don't code for any properties. I don't know if anyone's sort of using chemical or environmental methods to try to activate that mutation. I'm not sure about studies like that or aware of. but it doesn't mean they're not out there.
Can you use the DNA to say which parent, say a child has diabetes or multiple sclerosis, would the DNA say which parent gave it to them?
I would say that would sort of be under like clinical genetics.
Okay.
And the idea of doing, in a clinical setting, it would depend on the disease too. So how is it inherited? Is it something that's Mendelian inherited, where it comes from one individual or another? Some diseases are more complex, where it can't be teased apart what the source is. But if it's a simple hereditary type disease, then I believe I think that's what you go through genetic testing.
But, so if a child had let's use diabetes. If a child was diabetic, and neither one of her parents are diabetic, you're saying that the DNA from one of the parents, somebody that had them, or would they have given it to the child? I mean, would that way the child would have gotten it?
I would say I probably don't want to expand too much. I'm not an expert in clinical genetics or diabetes, so I'm not sure of the nature of that disease state itself. So the idea of, I think you'd have to know. I would have to know more, or if know more about the specifics about the disease state to know how it is inherited. Are there other sort of non-hereditary ways it can be brought and things like that? Yeah.
Okay.
Sure.
Interject with one more. These two are sort of related. In some situations, there is a lot at stake in the DNA tests. The tests seem very persuasive, but the chain of custody could be weak for the testing materials and process. The tester may be under pressure and may get sloppy. Anomalies like chimera may arise. How are these situations best addressed? And then in a second sort of related question, when random match probabilities are present in court, do they take into account errors in evidence collection, handling, labeling, analysis, and interpretation?
So I think for the latter question, when there is a random match probability, let's say it's 1 in a billion, that's not factoring in any of sort of the non-DNA related issues in terms of chain of custody or interpretation errors, things like that. So that's just taking that number, doing that calculation, and then providing that. So I'm assuming like in a law enforcement situation, they should be following a chain of custody and reporting system of how they're working. So, but the errors are not folded into, there's no uncertainty folded into the final estimate based on sort of laboratory technique or human error. Yeah. I think I talked about Y chromosome and mitochondrial DNA a little bit earlier. I just wanted to show like what we're talking about right now are autosomal markers, and those are passed on by all our ancestors. And you could see from the brother-sister example, as you keep going from generation to generation, things sort of get scrambled, or you get farther away from having matching between relatives. But there are these lineage markers can be useful, and one example here is the Y chromosome. So if you think about it, only males have Y chromosomes. And you get that Y chromosome from your father. So in this case here, if you have a son, he'll have the same Y chromosome as his father and as his grandfather. So if you tested the grandfather and the child, they would have the same Y chromosome. It's not saying that that he's the father, it's just that they're in the same paternal lineage. And just to note here too, if you look at your maternal grandfather, you're not going to have his Y chromosome. So if you're trying to do maybe like a genealogy or some type of lineage, and you're looking at your mom's grandfather and you're a male, you're not going to have his Y chromosome. You're only going to have your paternal lineage's Y chromosome. And then similarly, we talked about mitochondrial DNA, and the square here, I should say, is a male. So everyone has mitochondrial DNA, but it's passed on from your mother. So this son here, he gets his mitochondrial genome from his mother, and then she got hers from her mother. So the idea here is similar, that there's a specific chain of this type of lineage. So if you looked at your father's mother or your paternal grandmother, you would not have her mitochondrial DNA. And also too, you would not have your father's mitochondrial genome as well. So I'm not going to get into a lot of details with these, but sometimes these are used in studies where when the relationships become too far apart, like cousins or further or fourth or fifth generation, if you can put the lineage back together, that can give you some estimate of of identity, but usually it's a lower. You have to kind of take into account it could be any one in that paternal or maternal chain. So you're not going to get huge numbers like in the billions. But I don't want to say too much more about that. I just wanted to switch a little bit to what NIST is doing a little bit more in terms of our role in human identity testing. So of course, reference materials is one of these things, and this is to ensure confidence in results and support DNA testing. We do research on new technologies, and then we also have some training and education resources I'll talk about a little bit. So the main reference material we have here is a standard reference material, SRM 2391C, and that's the PCR-based DNA profiling standard. And I have one up here. This is from the FBI quality assurance standards, and it's a little bit dry, but it's number 9.55. But what it says here is that a laboratory shall check its DNA procedures annually, or whenever changes are made to a procedure against an appropriate and available NIST standard reference material or standard traceable to a NIST standard. So the idea here is any labs that are putting information into the database, the federal database, run the NIST SRM once a year, or if they get, let's say, a new kit or a new instrument, or they change their procedures, they're running the NIST SRM to ensure that they get the right answer. So this is kind of an idea to give you the foundation of the importance of this, or the user of this reference material. So this is what the reference material looks like. This is 2391C. There's 6 vials. And these are characterized, these have been typed and sequenced for those core markers that I've been talking about previously. So there's components A through F, and A through D are DNA samples. Those are just liquid samples containing genomic DNA. And then components E and F are actually DNA cells that are spotted on paper if someone wants to do the extraction. And then what we've done at NIST, sort of our value here is we took these genomic samples, we tested these on all the different markers and kits that exist for DNA typing. We've also sequenced them as well to see what those repeat motifs are, and that's what's in their certificate for 2391C. So when a laboratory tests this, and they're testing it to be for the QAS standard 9.55, when they test it, it's sort of calibrating their instrumentation, their PCR thermocycler, their techniques to make sure that they get the same answer that NIST gets with the SRM material. So it helps them calibrate their own instrumentation, and then it also helps ensure confidence between comparisons between different laboratories. So that was a bit quick, but this is something that's been going on for a long time. So what I kind of put up here is our forensic SRM timeline, and this is really just showing this series, this 2391 series, really started in the mid-90s or so. So we've been supporting the forensic DNA typing community since the early to mid-90s. And then the different iterations here, the 91A, 91B, 91C, whenever there's new markers or new tests that are available to that community, we update the materials in the SRM to support those. So one of the most recent things we did, I said that the United States is going from 13 to 20 markers. We characterize those other 7 markers, and now that information is in their certificate. So, and we have other materials out there. There's one for Y chromosome typing. There's one for quantitation, for calibrating your quantitation, and there's also ones for mitochondrial DNA as well. So we have sort of this suite of genomic DNAs that have been characterized at these genetic markers of forensic relevance. One area I was going to talk about was mini STRs. So I think, when we talked about the STRs, those different fragments, they can range in size from, let's say, 100 base pairs to 500 base pairs or so. So that's the size that you're looking at. Now, in something like a degraded sample, I think there was a question earlier, like what if you don't get the information off all of the markers? One reason for that is if a sample's degraded. So if you think of something that's been in the heat or high humidity, something where the DNA is fragmenting. So as the DNA breaks down, let's say you might have smaller and smaller pieces of DNA left over time from heat or UV radiation. So what NIST did was develop what's called mini STRs, and what those are the same type of markers that we've been talking about, but they're smaller sizes. So they're about 125 base pairs or so. So what they do, these work well with a highly degraded sample, and you would find a highly degraded sample sort of at, let's say, something like World Trade Center or a plane crash or some kind of mass disaster where things are undergoing a lot of heat and humidity and the DNA is breaking down, and under a normal test you're not getting full information. So we developed these tests at NIST in about 2005 or so, working with US industry, and some of this has been converted or passed over to companies like Promega or Life Technology. So an example I wanted to show here, this is some data that we're allowed to share from Bodie Technology, which is down in Virginia, and they were doing some of the fragments or the bone fragments information that they were getting from the World Trade Center in the mid-2000s. And this is an example of a test where the things I've circled here, the space out here that's sort of empty, this is where peaks are not showing up because the DNA is degraded. So you have, what I have here is over 12,000 bone samples were processed. About 3,400 gave full profiles, which were fine. And then you have this area here where you have these high partial or low partial, where you're getting about 7 loci or so. And then sometimes you get no information at all. But this is the case here where mini STR assays will sort of help. So the mini STR assays would really show up in this area here below 160 or so. So essentially what all these are a smaller PCR product size that works better with highly degraded samples. And then this has been really converted over to a kit called Minifiler that Life Technologies makes specifically for degraded samples. But this is an idea of how NIST played a role in the development of technology for typing degraded samples. Also, too, I mentioned multiple times now about the additional STR markers. So the U.S. is going from 13 to 20. What I have a list here is the left of the 13 that have been used since 1998. And these are the additional 7. And I just wanted to point out that the 2 markers here, D2 and D10, came out of that work from the mini STR. So NIST characterized these markers on population samples, published that information, and then those were actually something that were brought into the additional loci that the U.S. is going to be using. So just kind of showing our support or impact of characterizing genetic markers for the U.S. database. And then briefly here, something we're doing is we do kit testing at NIST. So whenever a company's coming out with these new kits, so they're coming out with their new 20-plex or 24-plex, they give us an alpha or beta version of that, and then we test those on about 600 or to 1,000 samples that we have at NIST to see if we have any discordance or incorrect genotypes. So the idea would be if someone has a new formulation, they give it to us, we test it on a bunch of population samples that we have all the information for, and if we find any discordances, we report that information back to the commercial vendor, and they can make any needed changes or adjustments to their kit before releasing into the forensic community. So the idea here is that by this kit, it's a little bit hard to see here, but there's 2 peaks here, I think a 13 and 15, but in the newer kit, it was just a 15-15, so you have a dropout of a peak here, and that could be adjusted by a sequence change in the primers that we talked about. I'm not going to get into this into too much detail, but just to state that this kit testing has been useful for supporting the development of these with the U.S. industry. So I'll finish up here with some of the ideas, the future of human identity testing. One thing I talked about before was rapid DNA, and we've done some work with rapid PCR protocols, but what these are companies, and there's 2 of them listed here below, Integenics and NetBio, that have these different instruments, and here's a picture of each of the instruments. And what this is for is really a swab in, profile out in less than 2 hours with no special training. So essentially, you could come up to someone, swab them, put it into this box, which has a consumable that can be thrown away after 1 run. You hit go, and then 2 hours, it'll generate a profile that's been analyzed for you. So one of the uses for this, you can think of multiple uses. One law enforcement use would be at a booking station. So if you want to enroll people in a database at the time of arrest, you can have this type of instrumentation at the scene, or not at the crime scene, but at the booking station. And when you fingerprint them, you can also swab them and enroll them into the DNA database as well. For DoD purposes, they could be used in the field for DNA testing, and possibly for DHS purposes for immigration or border crossing or human trafficking. So if someone says this is my daughter or this is my son, can you test them at the border and confirm that that's the relationship or not? But that's just kind of a high level of where this technology might be going. But the idea is that you could probably be trained to run this in about 15 minutes or a half hour or so to generate a DNA profile. And we have 2 of these systems at NIST that are supported by the FBI and DHS. And this is an example of a profile that was generated in less than 2 hours or so. And again, this is just putting a swab into a box and hitting go. There's no chemistry that the user has to carry out. So this is just the type of profile you get from an integrated instrument. I made a couple mentions to next generation sequencing. So, and since about 2005 or so, there's really these high throughput sequencing instruments that are really capable of doing millions or billions of bases per day. These are now commonplace, especially in clinical testing. So what I have here is something this is always being updated on the NIH website, and just looking at really this is the cost of doing a genome. So in 2000, it cost about $100 million to sequence a genome, whereas now we're at about just below $10,000 to sequence a 3 billion base pairs or so. So this is just kind of the drop off. This is right, of course, right about here, 2007 is when these instruments were coming onto the market and being used by researchers. So you can see really the kind of revolutionary effect that this has had on the cost of sequencing. And that's also driving what I have here to some non-forensic applications. So looking at clinical research, inherited disease. These are some of the questions that were being asked that can be sort of addressed with next-gen sequencing in a research setting. Looking at cancer, rare variants, transplant medicine. It really opens up, this level of sequencing opens up a lot more areas in terms of applications of DNA typing or DNA testing. I'm not going to get into these too much, but for our purposes, for my purposes, what would be the application for forensics? And the idea here is moving, you can still do STR markers, but now you can potentially do these SNP markers or these single variants that we talked about earlier. And there's been panels of these that have been identified in the research community for helping to predict biogeographical ancestry, or looking at things like eye color or hair color, or potentially complex kinship. And then you can also go into depth into the STR markers to see what the sequence is within that STR region. So there's a question earlier about what's the 22.2. By sequencing, you can know exactly what all the bases are in there. So really what I've been talking about is a length-based way of typing, the length of that product you associate with a allele size. But with sequencing, you might see some variation in the sequence that might give you finer resolution of the STRs as they exist now. So this is really some preliminary work that was done in our laboratory looking at sequencing some of the STRs. And this might be a little bit complex, but really what the blue bars are the number of alleles. This is sequencing about 200 people. And these are the blue is the number of alleles observed just by the CE, or the capillary electrophoresis, the traditional way of typing these samples. So the idea here for something like this THO1 locus, you might have 7 alleles that are detected. But by sequencing these, you get additional information that's not contained in just the length of that PCR product. So a good example here would be something like this D12 locus, whereby by traditional techniques you have less than 20 alleles or variants that are detected. But if you actually sequence, you find more variation within that STR, and you get above 50, or you get 36 more on top of that. So just the take home here is by sequencing some of these types of STR markers, we get finer resolution than we did by just looking at their length. I'll just show a little bit of data here. This is a kit from a company called Life Technologies or Thermo Fisher. This is their ancestry panel of SNPs. So this is a set of 165 sequence polymorphisms, and these were selected in the research community to estimate biogeographical ancestry. So we can run these on a sample. These are some of the papers that led to this work, and there's a database for searching them as well. And what I'm showing here is just kind of the interface of the instrument itself, where we typed a bunch of samples that were donated from George Washington University that had self-identified ancestry. So what the user might do is after these are typed in the instrument, and I'm not going to go through all the technology because of time, but clicking on the sample here, this individual, the code here is just S277. This person is self-identified from the Ivory Coast of Africa, so they were a student at GW. And after typing, you could go in here and click on the map, and it'll show you from that genetic information, it gives you an estimate of their sort of global ancestry, like where from their markers, from those SNP markers, where they may have come from originally. So this is an area that's of interest. You could think of like a cold case, where maybe you have a blood stain, you don't have any suspect. Can you type this sample and get an idea of ancestry? So this works. I think this works quite well in a global population, but sometimes it's a little bit less informative. Something like in the U.S., where you have lots of admixture and not such specific populations. There's also work out there for eye color and hair color prediction. This has really been led by a group in the Netherlands. This is a shot from their website. So they have SNP markers, these single bases that are used for predicting eye color and hair color, and these can also be put into a next-generation sequencing assay. And you would know the type from sequencing those, and then you'll have a predicting tool that lets you know is it blue eyes or brown eyes. So I've kind of pulled this out of the paper just to show that the level of accuracy for estimating these traits. So the idea here is about 70% for blonde hair, almost 80% for brown, 80 for red, and 87.5 for black hair. And then eye color is about 80% for brown and blue predictions. But if you have green eyes or something in between, it's not as accurate or so. But I kind of wanted to throw this up because this is sort of interesting to people to kind of take, again, sort of a cold case sample and see what kind of information can you get from that. But also stating sort of the levels of information you're getting aren't the 1 in a billion, 1 in 3 trillion like we talked about earlier. Because I think sometimes when people hear about DNA, in forensics, they assume that high level of certainty. So for these types of predictions, this is kind of where we're at right now in this 80% range or so. We're not sort of at that certainty of a match. And then finishing up here, you may have seen this recently in the New York Times. There's work going on with facial modeling, and what researchers are doing here are looking at different markers that might have something to do with bone structure, and then taking different individuals and sort of modeling their faces and sort of put together algorithms for predicting facial features. And we're not directly involved in this work, but this is something that you might see in the paper really more recently. Here's an article from February 23rd. In the New York Times, just talking about using this type of technology for predicting facial features, and how accurate is that is really the question, and could that be used in a meaningful way in crime cases? I think that's still, I'm not going to say too much, I think that's an area that's still being evolved and working in a research way. It's not really ready for casework yet. And then last couple of slides here are just some NIST resources. We have something called STR base. The address is up here. You can go to this website. All the talks from our group and all our papers are up here. You can download those at any time, and there's lots of different PowerPoint presentations if you want more information on STR typing. So this was created in the late 1990s, so you can go up here and find all our information we've kind of talked about for STRs, different software tools, the population data, things like that I've talked about, sort of the foundational information. We put it up here for free for the forensic DNA typing community. And you can also pull down any papers or workshops or presentations or talks that have been given in the last, since about 1998, 2000 or so. And then also too, I just wanted to show some of the textbooks written by my colleague John Butler at NIST. He was our previous group leader. So over the years, he's written different textbooks on forensic DNA typing, and these are used to teach in undergraduate and graduate courses, but also I find them, they're a pretty good read for the layperson to kind of understand DNA typing. I know judges and lawyers use these as well, because it kind of explains some of the different aspects of DNA typing in sort of easy to digest language. So with that, I'd like to just thank the group again, Mike Coble, Becky Steffen, Erica Rumses, Catherine Gettings, Kevin, Margaret, Jolynn, Dave, and Patty. They're all the folks that make this work happen at NIST, so I just wanted to thank them again at the end. And then some of our funding, of course, from NIST-based funding, and then work with the FBI, DHS, and NIJ over time. And there's my contact information if you want to reach me. I'm happy to take any questions, and thanks for your attention.